Hello and welcome. Today we're diving into one of the coolest tools in the entire forensic playbook. We're going to break down exactly how science can pinpoint a criminal in this explainer on DNA fingerprinting.
So let's start with the big question. How can something you can barely even see, like a single hair or a tiny drop of blood, tell such a huge story and basically solve a crime? That's the mystery we're unpacking today. Let's get into it.
Okay, so picture this. There's a crime scene. The investigators have gone over everything with a fine-tooth comb, but all they've got is this teeny tiny biological sample. We're talking maybe a few skin cells left on a doorknob. And that, right there, is a massive problem.
And here's the real challenge. They've got the culprit's genetic code, their DNA, but it's such a microscopic amount that you can't really do anything with it directly, you know? It's like trying to identify someone from a single pixel of their photo. It's just not enough to work with.
So, the whole reason this works is because of something called polymorphic DNA. See, most of our DNA is pretty much the same as everyone else's, but these specific spots in our chromosomes, they are wildly different from person to person. They're what give each of us our own unique genetic profile, unless you're an identical twin, of course.
So, how do scientists get around that not enough evidence problem? Well, they use this absolutely game-changing technique that's basically, and I love this analogy, a photocopier for DNA.
The official name for this magic machine is the polymerase chain reaction, or what you'll usually hear called PCR. And its job is brilliant in its simplicity. Take one tiny little piece of DNA and, in a lab, turn it into billions and billions of identical copies.
Seriously, the photocopier analogy is perfect. If you have one important page of a document and you need lots of copies, you go to a photocopier. If a scientist has one important strand of DNA and needs lots of copies, they use PCR. It's really that simple in concept.
Now, to get this photocopier running, you need a few key ingredients. You start with your original document, which is the template DNA from the crime scene. Then you need primers. Think of these as telling the machine exactly which part of the DNA to copy. And finally, you need your ink and paper. That's the builder enzyme called Taq DNA polymerase and the raw building blocks of DNA, the A's, T's, C's, and G's, to actually build the new strands.
Okay, so we've got our ingredients all mixed together. Now let's look at how the machine actually runs. It's this incredible three-step cycle that's all controlled by changing the temperature.
The whole process is just these 3 steps, repeated over and over. Step 1, denaturation. You crank the heat way up to 94 degrees Celsius. This is super hot, and it literally melts the DNA double helix apart into 2 single strands. Step 2, annealing. You drop the temperature way down to about 45 degrees. This cool down lets those primers we talked about find and lock on to their specific target spots. And finally, step 3, extension. The temperature comes back up to a nice, warm 72 degrees. This is the perfect temperature for that builder enzyme to get to work, adding all the building blocks and creating a brand new DNA strand. And just like that, you've doubled your DNA.
But here's the thing, you don't just do that cycle once. You do it again and again and again, usually about 35 times. And because you're doubling the DNA each time, the numbers get huge fast. You go from 1 copy to billions. Now that is enough DNA to work with.
So we've done it. We've made billions of copies of those unique DNA regions. But right now, it's just a bunch of DNA floating in a tube. How do we turn that into a visible fingerprint that we can actually look at and compare?
This is where we bring in a totally different technique. So remember, PCR was all about making copies. This next step, called agarose gel electrophoresis, is all about sorting those copies according to their size. The way it works is pretty clever. You load the DNA samples into this slab of gel that's kind of like Jell-O. Then you run an electric current through it. Now, DNA has a negative charge, so it naturally gets pulled toward the positive end. But here's the trick. The gel is like a messy obstacle course. The shorter, smaller pieces of DNA can wiggle through it much faster and travel further. The longer pieces get tangled up and move slower. And just like that, you've sorted all the fragments by size, creating a unique pattern of bands.
Oh, and one last little detail. DNA itself is totally invisible. So to actually see the results, a special fluorescent stain is added to the gel. It sticks to the DNA and makes all those little bands glow brightly under a special light.
And that brings us to the grand finale, the moment of truth. We can now put the DNA from our crime scene right next to the DNA from all our suspects and see what the evidence has to say.
And here it is, the final gel. Just look at the pattern of those glowing bands. You compare the lane with the crime scene DNA to the lanes from each suspect and, yep, there it is, a perfect, undeniable match. The pattern is absolutely identical.
Just so we're all on the same page, let's break down what we're looking at. Lane 1 over on the left is a DNA ladder. That's just for size reference, kind of like a ruler. Lane 2 is the big one. That's our crime scene DNA. And as you can see, clear as day, the pattern of bands in lane 4 from suspect number 2 lines up perfectly. Bingo!
So what's the big takeaway here? Why does this matter so much? Well, DNA fingerprinting is an incredibly accurate, statistically powerful way to tie a person to a place. And because of amazing techniques like PCR, you only need the tiniest speck of evidence to do it. This is the technology that fuels huge databases, like the FBI's CODIS system, that helps solve cold cases that are decades old and links crimes together to catch serial offenders. It's truly a game changer.
It's honestly incredible stuff, and it kind of leaves you with this one last thought to chew on. If just a few tiny variable bits of our DNA can tell such a clear story about where we've been and what we've done, just imagine all the other stories our full genome is just waiting to tell.